Hello all, welcome to the final week of this winter course on machine intelligence and brain research. Depending on where or when you are listening to this video, uh, good morning, good afternoon, or good evening. So we will talk about cell types that make up the brain in this lecture, and this hopefully brings in the different concepts that you have seen in the ML and neuro tracks of the course, and gives you a flavor of what we can do at the interface of neuroscience and machine learning. And my name is Manikandan Narayanan. I'm a faculty in the computer science department at IIT Madras, and also a coordinating faculty of the proposed Center on Multi-Scale Digital Neuroanatomy. So I hope to bring in the flavor of work that I do at the interface of computer science and biology to also to this lecture, and I hope you find it informative. Okay, so without any further ado, let's just get started. And let's start by first deconstructing this title. So what do each of these terms in the title mean? The neurotrack, the modern neuroanatomy 2, part 2. In part 1, you saw the circuits in the brain. And in part 2, we are going to look at the cell types of the brain. So let's just deconstruct this title a bit more carefully, okay? And before I jump into deconstructing the title and going further into the material, so I draw from different sources, both neuroscience and machine learning, ML. And I would like to thank all those sources, and I have acknowledged all of them in the bottom right of the relevant figure or text or slide that I have used. Okay, so having that said, let's just deconstruct the neuro track of the title. And of course, this course has been interleaving different threads from the neuroscience point of view and also from the machine learning point of view. And even though this talk is titled a neuro track, it will actually be an interface track, meaning that we will see some amount of neuro and some amount of ML. Okay, we will see both in this lecture. The neuro track would be about getting to know the different types of cells in our brain beyond the neurons, right? So neurons is, of course, the major players, and then you would have seen it either in this course or outside the course, pretty much a predominant cell type that you would see. But there are many other cells in the brain which support the functions of the neurons, and that is a glimpse of that is what we will see. That is the neuro track of this particular lecture. The ML track is modern neuroanatomy is relying on ML methods to analyze large-scale data sets to actually find these cell types, both neuronal and non-neuronal cell types, right? And so we would want to appreciate how machine learning is contributing to actually cataloging these different brain cell types and cell subtypes. So that is the machine learning track that comes in, okay? In a bit more detail or a bit more context, so the motivation for looking at it both of these ways is that once we have a understanding of the neuroscience or the neuroanatomy of the brain, so then that can eventually inspire new machine learning architectures, right? So, I mean, there is already a lot of connections between how the brain does intelligence and how artificial systems, artificial machines do intelligence, and there are brain circuit inspired artificial neural network architectures, right? ANNs. But they are mostly based on the understanding of a neuron or a model of a neuron, right? But can we also look at other cells that support the neuron in the brain? So understanding what these cells are and what they do, could they inspire new ANN architectures? So that is some out on the moon question, if you will. So can we, a moonshot question, if you will, in terms of can we use the knowledge of all the brain cell types that that exist to inspire new ANN architectures? And then the other part is this ML for neuro is can we understand better how the ML methods actually reveal what cell types and cell subtypes are present in the brain? And so that specifically means there is recent technological advances on obtaining single cell data at a very high resolution, molecular resolution, and unsupervised ML methods such as dimensionality reduction and clustering can be applied on these data sets to identify both neuronal and non-neuronal brain cells. Okay. And so how do we use these ML methods to obtain new knowledge on these different cell types? So that is the ML track. Okay. The neuro track is just getting to know the different cell types in the brain, and the ML track is about how does ML actually help in identifying these brain cell types in a data-driven fashion. Okay. So actually, we will be looking both at neuro track and ML track, and it's only apt. This comes towards the end of the course, and so you have seen many neuro and ML tracks together, and we wanted to show a flavor of both, blend them both into this last lecture. Okay. And let's now look at the second part of the title, the modern neuroanatomy too. To understand that, then we need to know what is classical neuroanatomy. And neuroanatomy is a field that is about 150 years old. And so this is based on this article in Cold Spring Harbor Labs. So initially, um, we all know about this pictures. Most of you may have seen these pictures by Roman Cahal, which was the intricate drawings of the neurons and their connections based on Golgi staining, right? So the staining that Golgi had introduced in 1873, right? And he was proposing a reticulate theory where he was saying that the brain was one continuous mass. All the neurons are not individual units, but the brain as a whole is one continuous mass that is interconnected. And Cahal took a closer look at the stains and then made these diagrams, several other diagrams, beautiful looking diagrams, and then said that there are these small gaps between these connections. And so they support actually individual neurons. So these neurons are some basic units, cells, essentially, that are then connected to other cells nearby, and they form this network inside the brain, the neural network inside the brain. And for these works, they were both awarded the Nobel Prize, and that is just one of the first recognition of neuroanatomy. And there are several other awards and recognitions in trying to understand the most complex organ in our body, in some sense, the brain, right? So another landmark was this Brodmann, who came up with this brain atlas. He divided the brain into different regions based on the anatomy of the brain, so anatomical structures, and associated some structural and functional organization, right? And then there were several other works, and then recently in the 2010s and 20s, the neuroanatomy field has taken a modern approach. By a modern, we mean a large scale, high resolution. So here you have these certain numbers, tens of 20s of regions that are being divided in Brodmann's atlas. They are still being very actively used, and this is still a reference atlas onto which newer data are mapped onto. But what newer studies are doing is taking a very higher resolution look at all the regions of the brain. So high resolution meaning you zoom in on one region and look at it in very much detail, but also large scale. You do that for very many regions. And multimodal, not just looking at how the neurons are connected, but looking at other cells as well, and how the neurons are connected to each other, how the other cells are connected to neurons, what is inside these neurons. So this higher resolution, meaning that not just at the level of cells and their connections, but also at the level of what are the genes and proteins inside these cells, and how can we use it to understand the brain's function better. And these all are coming together, these connectomes, single cell transcriptomes, looking inside the cell, connecting. Essentially, the next steps would be to connect these neural networks to the underlying gene networks that operates within each of these cells. And all of these are huge efforts, because you are not only zeroing in on trying to get a very, very high resolution on each region, but also doing it across all the regions of the whole brain. So naturally, it cannot be done by a single person or a single lab. And these are also consortium-driven projects involving multiple institutes and universities. So that is what modern neuroanatomy have evolved into, starting with single people making very deep contributions into a stage where large consortia are generating these massive data sets that tells you what the different parts of the brain are and how they are connected to each other in an unprecedented resolution. Okay. So that is the part about modern neuroanatomy. And the part 2 is about cell types. So we'll focus on cell types in this lecture. So in a week before, you hopefully have focused on circuits within the brain. So what is this cell types part here? So cells can be grouped. So these cells are the basic autonomous units in any living system. We are all made up of different cells, and they do the different functions in the body. And they can be grouped into different types, specifically cells within the brain, based on their structure, right, function, connectivity, how they look, how they appear, how their morphological characteristics are, and what function they perform, how they are connected to nearby cells, and their molecular state, what is inside these cells, what are the genes and proteins that are present within these cells. So you will see this word transcriptome time and again. It just simply means that what is the activity or expression level of all the genes in the genome within a cell. So that is called a transcriptome. So now I want you to stop or pause and think. Um, I have already told you that neurons are not the only cell types in the brain, but I would like you to, if it were an interactive session, I would stop and ask you to raise hands on who thinks the number of non-neuronal cells in the brain, what their guess is. And an even simpler question is, do you think that it is more than the number of neurons, or is it less than the number of neurons? So that's one question to stop and think about it. That will also tell why it is important to not just study neurons, but also other cells in the brain. And then can two neurons which appear very similar do entirely opposite functions? That will tell you that why we cannot use structure or appearance alone to classify cells into different cell types. You need to look at their function, their connectivity, or their internal molecular states. So I also want to pose this question that if two neurons which appear very similar, can they do entirely opposite functions? Okay, these are the two questions. I'll give you a moment of pause. And also to save bandwidth, let me just switch off my video. Okay. So let me answer these questions. So these are rough numbers. So the number of neurons in the brain are about 100 billion in our human brain. And the number of non-neuronal cells, these are the support cells which support the neuron in the brain, are about 1 trillion. So you can see that they are order of 10 more. And so it is very useful to know what these other cells are, what they do, because they are present in such abundant numbers, and so they must have some role to play. And despite neurons being the main signaling units, so it is they should get the prime attention, but newer research are showing that we should also pay more attention to these other cells. And then can two neurons which appear, excuse me, can two neurons which appear very similar do entirely opposite functions? So on that, I can say right away that there are excitatory neurons and inhibitory neurons. So neurons which activate other neurons to fire and neurons which inhibit other neurons to fire, and they may look the same in terms of their cell body structure, their axonal structure, their dendritic structure, and so on. So it's also important to know what their functions are and what their molecular state that enable them to do these different functions. Okay. So having given that context and motivation on why we should be looking at all the cell types in the brain, these trillions of cells in the brain. So what is a textbook view of things, right? So we have these neuronal cells. Like I said, there are excitatory neurons and inhibitory neurons, those which activate other neurons to fire and those which inhibit other neurons to fire. And then there are glial cells. These are these non-neuronal support cells. They support the neuron to do the various functions. There are microglia, astrocytes, and oligodendrocytes, among others. Okay, these are some major players of glial cells, major glial cells. And this material is taken from this particular resource. Neuronal cells, you would have seen, they come in wide varieties and shapes, different branching patterns, different diameters of the axon, different lengths of axon. This axon is this long fiber. They are insulated by this myelin sheet to conduct electrical impulses without interference, faster and also without interference from neighboring neuronal fibers, right? And you have the cell body which contains this nucleus and then the cytoplasm, the soma region, and you have these branching patterns, these dendrites. And of course, in these axon terminals and these dendritic terminals, you have these electrical signal exchange from one neuron to the other, these synapses. Okay, so this one you would have seen many times before, but just wanted to show you that there is such a diversity in even the shape and branching patterns and sizes of these neuronal cells. So maybe we should be even classifying them further into subtypes. Okay. And then these other cells, these glial cells, are the three major ones that I'm showing here. One is the microglia. The microglia is this immune cell, really. This is called macrophage in other parts of the body. And this immune cell does protect the brain against invaders, right? So if you have bacteria, virus particles, it can engulf them and then present it so that it can signal to other immune cells for help. And it can also do other processes, not just bacterial or viral pathogens, but also damaged neural tissue, damaged part cells in the brain. It can clear those debris up. So that is the role of microglia. So these are in some sense the cleaners of the brain, if you will, if I had to use a very crude analogy. And then oligodendrocytes are the ones which form these myelin sheets, right? So that helps in insulation of these axonal fibers. And and oligodendrocytes come from certain precursor cells called oligodendrocyte precursors. And so they develop into then full-blown oligodendrocytes that form this myelin sheath around these axonal fibers. And then there is astrocytes. These are called astrocytes because they look like stars. So they are star-shaped, astronomy, right? Astro. So they are star-shaped cells which hold the neurons in place, right? So you can see these feet. They call this end feet. They have these dendritic-like patterns, and then there are these end feet which hold both the neurons and also these blood vessels in place. And these blood vessels actually, more than keeping them in place, it's more for regulating the flow of blood, right? So it can control the flow of oxygen and nutrients through the blood vessels, and then it can also participate in some modulation of the signals between neurons. So that is some better appreciation of the role of astrocytes. Previously, it was thought to only do basically two major functions: one to support the neurons in place, and then to regulate this oxygen and nutrient flow in the blood vessels in the brain, which is crucial for a proper functioning of the brain. But now we know that they can also release gliotransmitters that can regulate how different neurons connect to each other. So even though this cannot in itself conduct electrical impulses, it can modulate the electrical flow through these other neurons. Okay, so for this reason, it's also called as a rising star in this a cell that is a rising star in this resource. Okay. So how did researchers discover or study these brain cell types? Okay, and seeing is really believing, and so they would basically using several techniques, but one of the predominant ones is called staining. You stain different marker proteins for these different cell types. So what are marker proteins? Proteins that are exclusively expressed or active in these cell types are those proteins that mark those cell types. So those are the marker proteins. They are present in exclusively or mostly in one cell type, but not in any other cell type. So then if you see those proteins where they are distributed in the brain regions, then you know where these cells are distributed. Okay, so that is the staining, and there are different types of staining: fluorescent-based staining, immunofluorescence they call it, and then there is immunohistochemistry-based staining using coupled with bright field microscopy. So there are different types of staining, but for the purposes of this lecture, I'm simplifying a lot of things and maybe even oversimplifying sometimes. But the flavor is to combine both the neuro angle and the machine learning angle, and so some simplification is inevitable. So, but with that, I will just say that you can stain these different marker proteins for different cell types. Okay. And let's see a particular example. This is from the source. I should have stated it here. This is from the the protein atlas, the human protein atlas. Okay. So there are these brain cell types that are shown here in a single picture. Previously, we used to show them as separate pictures, right? But now you are showing it in a single picture. N's are the neurons, O are the oligodendrocytes, A are the astrocytes, and so on. Okay. If you want to look at astrocytes alone, then there is this gene called GFAP that produces a protein called GFAP protein, and that GFAP protein can be stained, right? And then in this particular case, you can see these GFAP showing these astrocytic structures, or it's shown as brown over here, right? So it is shown as green over here, light green over here, and then it's shown as fluorescence over here, and it's shown as brown over here, right? And then it's shown in the schematic as this blue color, right? So all the other cells are not lighted up. But if you design the staining that will only stain GFAP, then the astrocytes are lighting up. Okay, as shown in these blue color, in these brown color, in this immunohistochemistry here, and then this is immunofluorescent staining, and this is immunohistochemical staining. Okay, and then there are also other things like oligodendrocytes. There is a protein called MBP that is exclusively expressed in oligodendrocytes, and you can you can again develop a method that will stain only MBP. And then you can see here that that is the only thing that lights up, and this is how it looks under a fluorescent stain. And this is how these brown colors, how it looks in a IHC, the immunohistochemical staining. And you can do the similar thing for blood vessels also. These are endothelial cells. SLC2A1 is enriched for expression. It's a marker protein for these blood vessels. And you can see here these different brown indications. And similarly for microglia, these immune cells which protect the brain against pathogens, you can use this protein called RGS10. So you can stain for this protein, and you can see here these little flashes of light green and these brown indications that mark these microglial cells. So this is how, like I said, seeing is believing. So this shows you that for different stains, you can look at the distributions of these different cells in different parts of the brain. But this, you may imagine now, or you may you may wonder now that are these perfect markers, right? So, I mean, is RGS10 only expressed in microglia, or are they also expressed at small levels in some other cells in the brain? Are these perfect markers? And if I want to understand not just any microglia, but I want to look at subtypes of microglia, maybe not all microglia are the same. There may be two major subtypes of microglia. Which protein should I look into to classify those cell types into even further into cell subtypes? So how do I increase the resolution of these techniques, right? To do that, we need to understand molecular biology a little more. And so that's what we will do next, okay? The overall question is that I want to be able to distinguish between these cell types and also look at sub-cell types, right? Cell subtypes. So within each of these microglia, there may be multiple subtypes of microglia. How do I look into them? And then how do I distinguish between those cell types and cell subtypes more clearly, okay? And a single protein may not be sufficient to do that. And this is where we need to get into the internal content of these cells, the molecular content of these cells, including genes, which are easier to measure than proteins at a single cell resolution. Okay, so to get to that, before we get to that, let's just get into some of the molecular biology of why these cells look so different. Okay, so there are all these different cells. We know that all the cells came from the same embryo. We have all the cells share the same genetic content, right? So all the cells within an individual, if I look at every cell within my body, they all have the exact same DNA. I mean, with some exceptions, there are some somatic mutations that happen, but for the most part, they are all identical DNA. And how does this identical DNA, all coming from the same embryo initially at conception, how did it all differentiate or diversify into such different things? I mean, some of them look like and form these tubes, some of them clamp here, and then some of them have very long axons, some of them are again like these myelin sheets. How does the same DNA produce all these different forms and functions and structure? Okay, that is the question that we ask next. Where does this remarkable cellular diversity come from, given that all cells within an individual have the same identical genome or DNA content, right? And the answer is actually quite simple. It's very perplexing that the cells all look very, very different. So that's amazing. But then the answer is quite simple in the sense that the subset of genes, so in our human body, for instance, we have 20,000 genes roughly, and then they all code into 20,000 corresponding proteins, let's say, but not all the genes and proteins are active in all the cells. If that were the case, then all the cells will look identical, right? Even though the DNA content is identical, the subset of genes and their products, which are these proteins encoded by these genes, they differ from one cell to another. And this difference in the gene or protein content of a cell, which I call it also as a molecular content or state of a cell, is what makes them different in structure, function, and so on. Okay. And again, all of these that I'm talking about has to be taken as kind of what is a predominant view. And there are always exceptions in biology. So exceptions are the norm in biology. And so for anything that I say here, like for instance, when I say all cells have identical genome, there's an exception. There are body somatic mutations that happen that will make one cell within me different from some other cell at certain parts. But for the most part, they are the same. And so just take these with these ever-present caveat that there may be exceptions in biology. But for the most part, like what I say here is the subset of genes and their protein products differ from one cell to the another. And that is what this change or this difference in molecular content is what makes each cell different and unique in their structure and function. And that's what makes them look so different. Okay. And so that is even further illustrated here. So living beings, here is a cartoon of one person, are made of cells. And then you take a look at one of those cells, there's a nucleus there, and then you look at the genome, the DNA content, the overall DNA content compressed into chromosomes within this nucleus, right? So that is identical from one cell to the other cell. But then these different parts of the genome, these are the different genes. So not all part of the genome are coding for genes. But a subset of them are. So here there are 3 genes which are being shown, these 3 wiggly lines, right? So three parts of the 3 regions of the DNA which are coding for 3 genes. In our human body, like I said, there is about 20,000 genes that the DNA codes. And then each one of them gets expressed. So this is the central dogma of molecular biology, where it shows the information flow from the DNA to the protein. So the proteins are the structures which do most of the functions within the cell. And then they are coded for by genes. And then these genes are encoded in the DNA, right? So the DNA sequence of letters forms the gene, and then that gene encodes the protein. And then so through an intermediary molecule called RNA, the genes get expressed, and then that gets through a process called transcription, and that gets translated into proteins through a process called translation, right? So let me just make sure that this is clear. Okay. So this part of the DNA is gene 1, let's say. This part of the DNA could be gene 2. And again, this part could be gene 3. So these are different substrings, if you will, of this long sequence of DNA. This is gene 3. And here it's only showing 1, but they need not be expressed at the same level. Some of them may be totally switched off. Other genes here may be switched off. There may be another gene 4, which is switched off. No expression of the gene or no activation of the gene is happening. But then gene 1 may be present in these 1 copy. Gene 2 may be present in 3 copies. I'll just do 3 RNA transcripts. And then gene 3 may be present in many, many copies. Let's say 5 copies. Right? So within this particular cell, so if I were to tabulate it, so this is a particular cell, cell 1. Sorry about that. So this cell 1, I have this gene 1, gene 2, gene 3, and all these other genes. I can tabulate them, right? and then there is gene 4 and so on. So I'm not showing gene 4, so I'm assuming that gene 4 is present in 0 copies. This is present in 1 copy, this is present in 3 copies, this is present in 5 copies. In some other cell, some cell 2, maybe gene 1 is present in 100 copies, and all these other genes are present in very few copies. Okay. And so on and so forth. Okay, so each cell has a different molecular content, gene content in this particular case, which also translates to different protein content because the genes through this intermediary RNA gets translated into proteins, right? So what I have drawn here is the number of RNA copies made of a particular gene, right? So that indicates the expression level of the gene. So the number here is referred to as the activity or the expression level of the gene, and then that also determines the level of the protein, right? So the gene content of the cell, which in turn determines the protein content of the cell, determines the function and the structure of the cell, because these genes, these proteins are the ones that eventually determine how the cell looks like, what function it does, what is the structure of the cell, what other cells it connects to, it talks to, and so on, right? So the proteins do all the functions of the cell. And again, this is a dogma. There are exceptions to it, but at the most part, this is what happens. And so this is what we mean by a cell is defined by its molecular content or state, and that is the central belief of molecular biology. There is another way to look at this. So, okay, you have shifted the question, where does the remarkable cellular diversity comes from? I have said that it comes from diversity in the molecular content. So where does the diversity in the molecular content come from? Again, we only have one embryo to begin with. And so how did it evolve into all these different cells, differentiate into all these very different cells? So where does the diversity in the molecular content come from? So we have only shifted the question. And that was attempting, a simple answer is that for each of these cells, the input conditions were different. And then they were allowed to evolve, and then they reached a certain steady state, which we call as a cell type, based on where they started off with. Okay, so Conrad Waddington said it way back in the 50s, I believe. So this is from another article which reproduces this figure, but this way before that, Waddington said, proposed this metaphor saying that this little ball here is like the cell, and depending on where you place it, it is going to roll down the cell and either end up in this straw or in this straw or in this straw or in this straw. Each of these straws are going to be different cell types. So this could be an excitatory neuron. This particular region could be an inhibitory neuron. These could be glial cells and so on. And really depends on the initial input conditions, where you place the ball. Do you place it right over here or to the left of here or all the way to the right here and so on, right? So, and these landscapes and where the initial conditions are all determined by this molecular content, which is shown as these little tethers connecting these little things, right? So these are the genes and proteins that dictate this landscape, okay? And as I said, this is just a metaphor. And so this leaves open for many of you who may be engineers who are trying to understand biology. This may be not too satisfying. But then this is a good metaphor, and it can be made concrete by using a dynamical system, basically a simple set of differential equations, ordinary differential equations. And let's look at a toy example here, two genes, X and Y. Okay. So the X and Y basically are two genes, and then this is in this flip-flop like system. Again, for computer scientists out there, you know that this is a way that you have a two negative feedback. So this bar says that X inhibits Y, or increased levels of X will cause Y to decrease, level of Y to decrease, and increased levels of Y will cause the level of X to decrease. Okay, so there is a negative feedback loop here between X and Y. And this and circuits like this happen all the time within the cell. So the genes in the cell are not working isolated. So all the genes are highly interconnected, like shown here. So one gene's activity can either switch on or off on other genes, and so they can either activate other genes or they can inhibit other genes. So there are either positive feedback relation among genes or negative feedback relations. And this is a very simple toggle switch model, or a flip-flop model, if you will, that is used for storing bits in memory when you think about it from an electrical point of view. Okay, but here this X and Y are genes, and then they inhibit each other's expression levels, and they are governed by the set of equations. So without going too much into the detail, what I wanted to have focus on is this phase portrait diagram. The phase portrait diagram shows X, the level of X, the expression level of the gene X on the X-axis, and the expression level of gene Y on the Y-axis. And at every point, it shows you how the X and Y are changing. So this dx over dt and dy over dt. Okay. I'm not sure if you can see these. These are little arrows, so they show both the direction of change and also the magnitude of change. The longer the arrows, it means the more the change that is happening. Okay. You can see here that this system cannot admit high levels of both X and Y because they will basically drive each of them down. One of them will go down to zero, to very low levels, and the other would stay at high levels. Okay, and those are these two attractors here. So if you place your if you start at any initial condition over here, right, and then follow the arrows, then you will slowly start here and then go towards this attractor. If you start at any other point over here, then the arrows again are very hard to see, but you will converge and slowly go towards this attractor. Okay, so this attractor is where Y is high and X is low, and this attractor is where X is high and Y is low. And these attractors in this dynamical system are really what cell types are, right? That are shown here as these troughs where you have the lowest potential energy. So this phase portrait can be converted into some kind of a quasi-potential landscape that is similar to this Waddington's landscape. Okay, so what is really the range of this landscape is all the values that X and Y could take, all the expression level values that gene X and gene Y can take. And then once you start at a particular point, then these equations here will lead you to a particular steady state, these attractor states. And these attractor states are really the cell type. So here there are two cell types possible, one in which X is high and Y is low, and then another in which Y is high and X is low. And one could be an excitatory neuron, for instance, and another could be an inhibitory neuron, for instance, right, in this toy example. Okay, so I hope that gives you some engineering point of view, as I understand that many of you may be engineers who are listening to this lecture. So what we would like to do is move beyond these toy systems and see if we can use the overall expression of all the genes in the genome. So the global gene expression. Okay, so can we classify cell types based on this modern global gene expression view? And essentially, this textbook view or this protein expression view that we had of these neuronal cells and glial cells, can we further subtype them? Can we distinguish them more carefully by looking at all the genes within these cells? Okay, that is what modern neuroanatomy does. It tries to see if we can revolutionize how we identify cell types and subtypes that make up brain regions in a large scale data driven manner. Okay, that is the goal of how modern neuroanatomy have evolved to identify cell types and cell subtypes. And then how you do that, the answer is to look inside a cell. Okay, so the question is, can we discover them using a data driven fashion, these different cell types? And the answer is to looking inside a cell by looking at its molecular content, and not just one or two genes, but the overall set of all the genes in the genome. Okay, and that, as I said, is called the transcriptome. It is the expression level of all the genes, the activity level of all the genes inside a cell. And it is single cell because you're looking at it within each cell, not like looking at an average of a bunch of cells, but looking at each cell and looking at what genes are present within each cell. And then this is a huge data set. And that obviously then means that we need computational methods, machine learning methods, to help cluster such data sets, the cells cross genes matrix, to find cell types and cell subtypes. Okay. So that brings us to a particular transition now. So we have looked at all so far, the different brain cell types, the neuronal and the glial cells. And among the glial cells, we have looked at microglia, oligodendrocytes, and astrocytes, right, which do different functions within the brain. Now we would like to know whether we can further subtype them, and then whether we can understand the genes that are behind these cells, these distinct cell types better. Okay, and in a data-driven fashion. So which means that we need to know what the data is, and that is where the single cell transcriptome comes in. Okay. Okay. Like I said, let's look at the data-driven fashion. So to do a data-driven classification of brain cell types or clustering of brain cells into different cell types, let's look at what the data is very briefly. Okay. And then we will go on to the ML methods that help you analyze such data to identify cell types and cell subtypes. So that is the agenda for the rest of the lecture. And finally, use a case study to show why we do all this, how it gives you new insights about a particular brain disease, neurodegenerative disease, which is the most common form of dementia called Alzheimer's disease. Okay. Okay, let's get into the brief look at what the single cell transcriptome data is, which is this global gene expression of an individual cell. So the predominant way before the single cell revolution came about in the last several years, the predominant way of understanding a brain tissue or any other tissue for that matter was bulk RNA-seq. So RNA-seq is a technology for measuring the RNA content of a cell. And so because I said in the central dogma that genes get translated into proteins through this intermediate RNA, that tells you the expression levels of the genes by measuring the RNA. So RNA sequencing gives you it measures the expression level of the different genes. But it was typically applied traditionally in a bulk setting where you take a tissue sample, you ground it all up to extract the RNA from all these cells, and then you measure. So you have millions of cells in a sample, and then you measure all of that. All you get is one measurement of each gene in this sample, which is the average across all these cells. So that's an average measurement. But then what the single cell technology has revolutionized is that you can dissociate this tissue into individual cells, and then you can actually peer into each of these cells to know what the gene content is within each of these cells. So that is a part about the single cell analysis where you separate these cells out into different small wells, pico wells, or into different droplets, nanometer sized droplets. And then within these droplets, you do all the necessary experimental reactions to enable measurement of the genes within each of these cells. Okay. And so here, for instance, you are seeing that this red gene is only present in the fourth cell, and this dark blue gene is present predominantly in the first cell and not as much in the other cells. And so once you utilize this the different genes measurements, these different genes expression levels in each of these cells, then you can plot them up after applying some ML methods to look at this reduced dimensionality visualization, and you can see these 4 distinct cell types. But then you cannot do such a thing. You will just have one data point for your average measurement. So you cannot really understand the variation from cell to cell and what the types of cells are and what the subtypes of cells are. That will be very difficult to do here, but in here you will be able to do it. And this is where your ML methods are applied, right? To take this particular data, which is just another way of the cells cross genes data, right? So you have these cells, these 4 cells, and then if you want to look at all the genes, these are the genes. And in cell I and gene J, this ijth entry will tell you how many copies of gene J is present in cell I, right? So this is, then you apply a transformation on this to get this particular visualization of the data or clustering of the data into cell types and so on. And this single cell aspect of this and RNA-seq aspect of this involves many other sub-steps, right? That we will briefly show now before illustrating why the average is not always a good idea. Okay, I mean, this is already, I hope, clear here. Why average gives you only one data point, but then here for every cell you have a data point, and so you can look at how the cells are distributed, what is the variation from cell to cell, and how you can group them based on that variation, right, into cell types. Another way to look at the average is that average is sometimes good as a representative of a population. If you want to do a histogram or a density plot of a gene, right? So this is the expression of a gene. This is the expression level of a gene. I will call it gene expression, right? And then this is the density plot. So this is the frequency of the density. across all the cells. So there are many cells which are in this region with this level, maybe 10 units of this gene expression. And some cells are with some 5 units, and then some others are with 15 units, and so on, right? In this particular case, the average is 10, and so 10 seems to be a representative of the population of all the cells here, right? So there are some cells over here, some cells over here, and so on. This is just for one particular gene, just for simplicity. But consider some other scenarios where you don't have a normal like bell curve, but you have, let's say, a more heavy tail. So maybe the gene is present with a particular mode, but then there is a heavy tail here, that there are many, it's not symmetric, and there are many cells with also higher levels of expression of this gene. So this is for gene G1. Let's say there is some other gene G2. Right here, the average is going to be somewhere over here, and then it will be representative of the cells over here, but then not really of the cells which are present all the way to the right here, right? So the average is not representative of this. So this is a standard issue with averages, and there is even more one that's where the average is not representative of any cell at all, right? Which is when you have multiple modes. When you have a particular gene which is present in low levels in one set of cells, and then a high level in another set of cells, right? So then you have these, let's say, excitatory neurons, and then these are inhibitory neurons. The average is going to be somewhere in here, which is, there is very few cells in this region, and then it's not representative of either this set of cells or this set of cells. So this is really a bad situation. This is a bad situation. Here it may be okay to use average as a representative, okay? So this is why looking at each of these individual X's is important rather than just looking at this one single average, okay, which is a very crude approximation of the population. But also in some cases, it's not even representative of the population, like in this case where there are two modes, okay? Okay, so this scRNA-seq stands for single cell RNA sequencing, that tells you the expression level of genes within a single cell. In some cases where the cells are difficult to separate out, like in neurons with long axons and dendritic branches, you also do snRNA-seq, which is single nucleus RNA-seq. For simplicity, I'll just mostly use the word scRNA-seq. It will also include single nuclear RNA-seq, okay? And that is a very long process. So whatever I showed here in this step, single cell analysis RNA-seq, it can be expanded out into a long process. And then we will mostly deal with this genes cross cells abstraction, okay, that matrix. But then I want you to know that single cell data is very noisy. So you'll need to know when the variation from cell to cell is biological and when it is technical or noise, right? And to do that, you need to be able to understand some amount of the upstream processing. So that is beyond the scope of this lecture, but there is excellent material here on the single cell course that you can refer to. And this picture is taken from that through this original article here. It shows you how the initial sample preparation is done on separating out the cells from a complex tissue, then feeding it into one of the ways of measuring the single cell. It's called a droplet technology. There are other technologies as well, but these are droplet technologies where within a small oil droplet, you capture each individual cell, and then all the reactions happen within that droplet. Then you do your library preparation to do your sequencing. And once you have the sequencing, then you have to do the separation of that into the different cells and the different genes. All of this process are important. But as I said, it's a scope of a different course. And what we will deal with is, it is important to understand so that you know which genes to keep, which cells to keep, which ones to filter out. So there are certain important quality control steps you have to do before you can start using the data. But we will assume that all of that has been done, and we are given with a clean data that has cells cross genes. Okay, so in each cell, N cells, you have M genes. So in humans, it will be like 20,000 genes, and then cells will be using these droplet-based technologies in tens of thousands, right? So you have N cells cross M genes counts matrix, and we will also, it is initially in the form of integer counts, and it is appropriately filtered. Bad cells are thrown away, bad genes are thrown away, and then appropriately transform, log transformation, for instance, is used. It has many zeros in this matrix, so that is what I mean by noise. Many genes are at low levels, may not be detected by this technology because each cell has such a minute amount of RNA to start with. And so you have to transform the data appropriately to get this final matrix, okay? And what our focus would be on this really this last step, and that too just on the clustering part, okay? How do you get this matrix and then try to understand this matrix so that you get group these cells into different cell types, okay? So that is, we are going to use something called spectral clustering, and I will shortly tell you why I chose this, because it serves kind of two purposes, like so two mangoes with one stone, if you will. And the reason is that there are two unsupervised ML problems that are typically applied for single cell data analysis. One is dimensionality reduction, another is clustering. First of all, just to briefly remind you, the unsupervised machine learning is recognizing patterns in a data set. By a data set, I mean set of n data points. We have n cells here, right? Each of them is in R to the m. It's an m-dimensional vector, right? So the values of the m genes within each of those cells, right? So these are real numbers. So there are m such real numbers, m tuple of real numbers that define a particular data point, and you have n such data points, n cells and m genes, right? And you don't have any labels to begin with. That's why it's an unsupervised problem. You do not know whether this cell is a neuron or this cell is a excitatory neuron, or this is a glial cell, or this is a microglia, and so on. So you don't have any such labels, and you want to cluster this data. So that is this that is this cluster, grouping n objects into k clusters based on their similarity. Like within each cluster, the objects should be more similar to each other than one object in one cluster and another object in a different cluster, right? And then dimensionality reduction is about reducing from this very high dimension m. So it may be in the thousands. So you initially start with 20,000. After some filtering and other things, let's say come down to 8,000 or 5,000. That's still a huge number to visualize the data. Can you bring it down to a smaller number, like 2 or 3, so that you can visualize all the cells in paper in a computer screen, right? So how do you transform your data points from very high dimensions, m in the thousands, to low dimensions, m in the 2 or 3 or maybe tens, without much loss of information, right? Assuming that and of course this has to be based on some assumptions. You cannot just take any arbitrary data set and reduce dimensionality without losing information. Assuming that the data points lie effectively in some low dimensional representation of the original space. And why is that possible for single cell data? Many of the genes, these 20,000 genes, are not independent of each other. Like I already said, these genes are all tightly connected to each other in these gene networks. And so many genes are correlated to each other. And so even though you have 20,000 to begin with, some of them may not be expressed at all in cells. And then some others may be highly correlated into groups of genes, which act as a single module of genes. And so there is hope that you can bring down the number of dimensions. And in both of these unsupervised machine learning tasks, there are many approaches possible. So PCA, principal component analysis, t-SNE, UMAP, and so on. And Laplacian eigenmaps is a particular idea based on the spectrum of a matrix. So spectrum refers to the eigenvalues of a particular matrix. And we will choose to look at it because, again, there are many approaches to clustering, like k-means hierarchical clustering, k-means clustering, and spectral clustering. And spectral is something that if we learn that, then we learn both dimensionality reduction and clustering using an example method. This is by no means this only method that is being used. In fact, t-SNE, UMAP are more popular for single cell analysis. But then in the particular application in this Alzheimer's disease analysis, they use some form of spectral clustering and some of the methods that we look at. And so because it covers both dimensionality reduction and clustering, I thought it will be a good one to look at. And you may have in your engineering curriculum seen PCA or k-means or other simpler clustering algorithms before. So that is another premise with which I chose to focus on spectral clustering. Okay. So before I go to spectral clustering is a graph-based method. So I need to first convert my data matrix to a similarity graph. Okay. What do I mean by that? The data matrix is n cells cross m genes, right? So n cross m. I want to make it into an n cross n. So where the nodes of the graph are the cells, And then edges of the graph indicate how similar two cells are. Okay, if you think about it as a complete graph where all the pairs of cells are connected to each other, so we have n squared pairs, then the cell I and cell j, this xi is this m-dimensional vector, xj is this another m-dimensional vector that refers to the gene content of cell I and cell j, right? You can use different similarity functions. One choice could be that you look at the Euclidean distance, squared Euclidean distance between the two. This indicated this norm of xi minus xj squared, and then divide by some parameter sigma, 2 sigma squared. Okay, sigma is a parameter which you can tune it, or you can use any other similarity function. The only requirement is that it has to be non-negative. Okay, and higher the number means more similar the cells are. And this particular example function will satisfy that. It basically converts a squared distance, squared Euclidean distance, using this e to the minus that distance divided squared distance divided by 2 sigma squared into a similarity, so that this S is always non-negative, 0 or higher. And this can be defined for all pairs of cells, xi and xj, okay? But then oftentimes we don't want to work with a huge similarity graph that has n squared edges, right? You may want to work with sparse graphs. And so there are other ways of converting. So if your data point really lies in only 2 dimensions, so here m is 2, just for simplicity, let's say m is 2, then if you want to construct a sparse graph, you can look at 2 different approaches. One is called epsilon neighborhood approach, where you fix some kind of epsilon to be 0.3 in this particular case. And then you say that I will connect every cell here, every dot that is shown here to all the cells within a ball of radius 0.3 around me. Okay, so so that is this epsilon neighborhood. Whichever other cell is in my neighborhood in distance at most 0.3, I will connect it. And others I won't connect. So you will see that there are these little light green edges which are connecting those which are less than 0.3 distance apart. Okay, and then this KNN graph, but then here it, of course, you need to know what this value is. 0.3 will give you this, 0.4 will give you something else. And then a priori for 2 dimension, it may be easier to figure out what this parameter epsilon should be. But for high dimensions where m is really high, in our case in the thousands, it may not be as easy to determine that. One in which is relatively easier to set up is this k nearest neighbor. You just connect every cell to its k nearest neighbors. So you look at the distance to every other cell, and then you look at the, in this particular case, 5 closest cells, and each cell connects to the 5 closest cells. So here you can try again, k is still a parameter, you need to figure that out, but this is less sensitive than this parameter epsilon. So you can it's a bit more easier to figure out what the k should be. And in fact, if you do one more thing, so here you can see that all the dense regions are connected, but sometimes 2 different regions also appear to be connected. If you use something called mutual KNN, where 2 things are connected, if 2 nodes I and j, or 2 cells I and j are connected, if I is within the 5 closest neighbors of j, and j is within the 5 closest neighbors of I, right? So then some of these connections which link between 2 different looking clusters will not be connected. Okay. In the end of all of this, you are converting your single cell data matrix n cross m into a similarity graphs adjacency matrix. It's either a complete graph with every entry I and j being non-zero, or it is a sparse graph where many entries are 0 and then some entries are non-zero according to these schemes. Okay. Once you have done that, you have this this adjacency matrix A. So this is this n cross n similarity matrix that I was talking about. It's, in this case, it is sparse. Let's say that this is your original data. I mean, none of the single cell data may look like these nice concentric circles. It's a toy data set. So you have this green cluster, you have this blue cluster, and you have this red cluster. Simple methods like k-means clustering won't identify these 3 clusters for various reasons. But the focus here is not on k-means clustering, but more on spectral clustering. How will it perform here? Okay. Spectral clustering is really 2 steps. One is dimensionality reduction, and another is some other simpler clustering methods such as k-means. So if you don't know about k-means, that is okay. You just assume that it is some simple clustering method that is quite easy to implement, and that given a data set, it will find out what, and given a number k, let's say k is 3 in this case, it will find out the 3 clusters. It will group the data points into 3 clusters. Okay. So the dimensionality reduction is basically shown here. Okay, you have this n cross n matrix. Initially, this is in an n cross m space. M is 2 here, just for a toy example, but m is usually in the thousands. So from n cross m, you have come into n cross n similarity matrix here in this matrix A. Then you do some dimensionality reduction over here. Okay, so this is where you are doing dimensionality reduction. And you get this n cross 3 matrix. Okay, so this is an n cross n matrix. This is your n cross m data matrix. And then here you have come down to n cross 3, where 3 is the number of clusters, n cross k. Okay, now if you visualize it, if you do this dimensionality reduction using something called Laplacian eigenmaps, you will see what that is. Then you will get these nice 3 clusters. Okay, so this green cluster, this red cluster, and this blue cluster. Okay, and then if you apply k-means, it will nicely group these together as this is one cluster, this is another cluster with its cluster centroid, and then this is a third cluster. Okay, so these could be, for instance, one could be microglia, another could be a neuronal cell, another could be a astrocyte, right? So this is the overall idea of spectral clustering is, and that is also why I said it does. I mean, if you know spectral clustering, if you learn spectral clustering, you have learned both dimensionality reduction and clustering in one go. Okay, that is why we also chose it. Okay, so how do you get this done, right? So I mean, this dimensionality reduction is one step and clustering is the other step. The clustering part, we will just say that some simple clustering algorithm like k-means could be applied. So we won't go into it in detail, but we'll just go into this dimensionality reduction part a bit more. Okay. So let's just look at what is the problem that spectral clustering is really trying to solve. So you have the input, which is the similarity matrix. Aij is the similarity between data point I and j. In our case, cell I and cell j. Each one is an m-dimensional vector. And so this similarity matrix is an n cross n matrix. Each one of them is 0 or above 0, right? So this is a non-negative n cross n matrix. We want an output which is in some kind of score assignment to all the nodes, right? U, which is an n-dimensional vector. So every node you have a particular value that you give it, so that this objective function is minimized. Okay, this loss function is minimized. Okay, and this is not really the objective function. We'll get to it very shortly. We will just say this is an approximate objective function. That's why I had this tilde sign before. But let's look at this objective function and try to understand this. Okay, this loss is given as 1/2 sum over all the pairs ij, right? All the pairs of nodes or all the pairs of objects or cells, whatever you want to call it, aij ui minus uj whole squared. Okay, so A could be like this. So this could be the graph where you have two triangles, right? And then the adjacency matrix would look like this. The first 3 nodes, 1, 2, 3, are shown here. And the next 3 nodes are shown here, right? And then the weights of these edges are all 1. The similarity Sij is 1 for all of them. And if there is no edge, then the weights are 0. Okay. And basically, you can think about how to minimize this. So one trivial solution is that you make all the ui's and uj's 0. Right, so score assignment to all the nodes, make all of them 0. So then it's 0 minus 0 is 0, and then this whole thing becomes 0. And this whole thing is aij is a non-negative matrix. Ui minus uj whole square is also non-negative. So this L of u is non-negative. It is 0 or above 0. So then 0 is the lowest possible that can happen. So that is a trivial solution. So let's go beyond that. Let's say that I don't allow trivial solutions such as all 0 vectors. Still, there is another trivial solution where I can make everything to be a, the same value a, okay, some constant value a. This is also not a very good solution because it doesn't tell me what the cluster structure is, right? If I have all of them to be a, then ui minus uj is again a minus a, 0 for any I and j, right? In this case, there are 6 nodes here. And then that will also minimize this L of u and will make it 0. And so that is also not an ideal solution. We will fix it shortly later. We can avoid this this trivial solution by ensuring that by enforcing that the u's are all eigenvectors of a particular matrix. Okay. Again, I'm assuming some form of. I hear that many of you are engineers who are in the course. So I'm assuming some form of matrix algebra knowledge and linear algebra knowledge here. If you do not know that particular prerequisite, that's okay. You can just quickly go through these set of discussions, and we will come back to how this all connects to brain cells and brain cell types, okay, in terms of the applications. So please bear with me if you are not aware of these terms. So, but by enforcing some, by looking at a particular related matrix called a Laplacian matrix and looking and asking or enforcing that U has to be eigenvectors, orthogonal eigenvectors of that matrix, the second part can also be ignored. And the main thing over here is we want a U that looks like this, where the first three nodes here, it looks like there are two clusters, one triangle and the other triangle. The first three nodes get the value A, and then the second three nodes get another value B that is different from A, right? So that when you look here now, whenever Aij is small, that is those connections between the clusters, Aij is 0 here, right? So for any I in one cluster and Ij in the other cluster, there is no edge and all of them are 0. So then for those Aijs which are 0, Ui and Uj can be anything. In this particular case, it will be A and B, right? But for those Aijs which are high, which is for I and j which are within the same cluster, like here between 1, 2, and 3, or 4, 5, and 6, right? We want the U and Uj to be the same as possible. So in this particular case, it will be A minus A or B minus B. Okay, so I hope that is clear. So L of U will be small if Aij is small, in which case U and Uj can be anything, right? Because all you have control over is U. You don't have control over Aij. That is provided as an input to you. You need to figure out what U is. So as long as you assign the same score U to members of the same cluster, then if Aij is high for members of the same cluster, Ui and Uj will be more or less similar, and they will be then close to 0, and the whole term will come down to 0. Okay, so that is what, so we want something that looks like this. Cluster 1 is given all some constant A in the U vector, and then cluster 2 is given all some other constant B. Okay, that will be the ideal solution. In fact, instead of asking for one U, I can also ask for two different U's, right? And then I can say that another solution here would be that for cluster 1, it's an indicator vector. I can say I want 1, 1, 1, 0, 0, 0, or A, 0, 0, 0. And another thing could be 0, 0, 0, B, B, B. Right? So then this will be an indicator for cluster 1, and this will be an indicator for cluster 2. Right? This will be one particular U vector, and the other thing will be another particular U vector. So instead of this A and B both combine into one vector, I can also ask for two different vectors where each vector reveals immediately what the clusters are, these two triangles. Okay. So that is the intuition behind spectral clustering. We won't go into all the math behind it, but we will just point out the main intuitions. One more derivation that we will do is this. I told you about how these are some trivialities like this all being the same value A can be avoided by enforcing this eigenvectors restriction on U. Let's just look at it a little bit more. This L of U is this particular expression. We just reasoned out when that is small. You can now expand it out by saying this is ui squared plus uj squared minus 2ui uj, right? And then if you now gather those terms, it's using simple algebra. You will see that L of U is, there is this minus 2 ui uj, and then it cancels with the half, and you will get this sum over ij, ui uj minus aij, and then you have this other term, which is this ui squared plus uj squared. So that again comes twice, right? So that 2 and 2 gets cancelled, and then you will get basically this ui squared into sum over j equals 1 to an aij. And what is this? This is simply the degree of a node, like the sum of all the edge weights incident at a node. In this particular example, you have this as 3, right? This is basically the row sums or the column sums of this matrix. Okay, so this D is a diagonal matrix where along the diagonal you have the row or the column sums. This is also called a degree matrix because degree is basically the number of neighbors or the sum of weights of the edges from you to all your neighbors. Okay, in this particular case, it is 3 because it's simply the row sum or column sum. So then this is your D here, right? And then this is your minus A here. So then if you combine both of these terms, then you have a vector U transpose of that U transpose. You have a matrix which is D minus A, where D is this diagonal matrix DIA given by the degree along the diagonal entry. The row sums or the column sums, right? And minus A. A is a symmetric matrix. That's why, similarity between I and J is the same as similarity between J and I, right? So that is why you can either look at it as a row sum or a column sum. Both will give you the same answer. So you have D minus A times U. So what we have expressed is this particular intuitive objective function that we had reasoned out on when it will be small, that will reveal the clustering structure of the data, underlying graph, is expressed as this U transpose some matrix D minus A times U. And this matrix is called the Laplacian matrix or the unnormalized Laplacian matrix. And so this D minus A matrix, then if you look at eigenvalues and eigenvectors of this L matrix, then that gives you this desired U that we are looking at. Okay. And why is that? And before looking at it, let's just understand to make sure that everyone is on the same page. So this is your similarity matrix A for the similarity graph shown here. So this is your similarity graph. I'm not showing the self loop. Each node is connected to itself with the edge of weight 1. I'm not showing that, but each of these edges is weight 1. Right, and each of these dots are the nodes 1, 2, 3, and 4, 5, and 6, right? So you have the node 1, node 2, node 3, the first triangle, the node 4, node 5, node 6, the second triangle. So this is your 3 cross 3, n cross n matrix, 6 cross 6 matrix. The D is again your n cross n matrix with degree 3. So L is going to be D minus A. So it will look like 3 minus 1 is 2. Then this is going to be minus 1, and then you'll have 0, 0, 0. Similarly, you will have minus 1, 2, minus 1, 0, 0, 0. And then you will have minus 1, 2, 0, 0, 0. Then you have 0, 0, 0, 2, minus 1, and so on. Right? So you have this block diagonal form. You have this block here, and then you have this block here for cluster 1. All of this is 0. Okay. Now we can see here that if for those of you who remember or know about eigenvalues and eigenvectors, so a vector X is a eigenvector for a matrix A if AX is some constant lambda, the eigenvalue times X. Okay. So the operation by the matrix on the vector just simply scales the vector by a factor lambda. So you can see here that for this matrix L, this Laplacian matrix, you can see that an eigenvector U would be something like all ones vector would be an eigenvector, or these vectors are all eigenvectors. So this is an eigenvector. I will call it U1. U2 is another eigenvector. And there is another U, which is the addition of any of them is also another eigenvector, all ones vector. Right, all of these are eigenvectors with eigenvalue lambda being 0. Okay, so these two vectors are orthogonal to each other. They are perpendicular to each other. And this is just a linear combination of U1 and U2. Okay, so you can see here that for this, you can just verify it easily, right? So you have 1, 1, 1, 0, 0, 0. So you just multiply this L with U, L U1. Then you will see that this 2 minus 1 will get added up, and that will be 0. This 0, 0, 0 doesn't matter because this is 0, all zeros, right? So L U1 will be 0. So you will have L into U1 vector would be 0 into U1 vector because that is 0, right? And then L into U2 will be similarly 0 into U2 and so on, right? So so u1 and u2 are perpendicular to each other. Their dot product is 0. And then so they are both eigenvectors of this eigenvalue 0. Okay, and then you may have eigenvalue 0 is just one of the eigenvalues for this matrix. You may have other eigenvalues for this matrix L also. Okay. And just to make sure that this is not confused with this other A, so I'll just put this as L. The matrix Lx equals lambda x. Okay. So is it just a coincidence that the 0 is a small eigenvalue? I mean, 0 is the smallest non-negative number you could get. And you could show that this L cannot have any negative eigenvalue. So in that sense, 0 is the smallest eigenvalue of this particular matrix L, Laplacian matrix L. And this seems to indicate the clustering structure. This first triangle is indicated by U1, where all the members of the first cluster are ones and everything else is 0. And U2 seems to be indicated by the indicating the second cluster, right? It's the second cluster is all 1, and then the first cluster, other entries are 0, right? Is it a coincidence that the smallest eigenvalue 0 corresponds to these kind of indicator vectors for these clusters? Or is it true for other examples as well, right? Or is it only true for this example or also for other examples? So we're not going to go into the theoretical aspects of it, but you can show that if you have a graph where you have these different connected components or these different pieces, then each of these pieces can be indicated by one of these eigenvectors for the corresponding eigenvalue 0. And the multiplicity of this eigenvalue 0 will be the number of connected components in this graph. Okay. If these terms are new to you, you can just a take-home message will be that small eigenvalues and the corresponding eigenvectors will indicate what the clustering structure in the data is. Okay, let's look at it a little bit more. This will be the kind of the last but one slide on the technical aspects of spectral clustering. Let's look at all the eigenvalues of a particular matrix. It could be for a data set that looks very clean, like these two triangles, or it could be a data set which has some more noise, that there are two clusters, but there are also some edges that cross the clusters. It's not like fully disconnected. Okay, so there are some edges that cross the clusters, but within the clusters, there are many more edges than there are between the clusters. Okay, so let's look at such a similarity matrix and then compute the Laplacian of that and compute the eigenvectors. And arrange all the eigenvectors. You can show that all those eigenvalues should be non-negative, and arrange all the eigenvalues from smallest to largest, and look at the top a plus 1, like the smallest a plus 1 eigenvalues. Okay. Now for any such eigenvalue lambda a, lambda k, between lambda 0 to lambda a, or any, and the corresponding eigenvector uk, right? So you will see that this uk transpose L uk, we have already seen that this is the same as the loss function that we want to minimize, right? That we have seen in this previous slide, right? So we have seen that this L of u, which is this objective function that we want to make it small to reveal the clustering structure, is basically this u transpose Lu, right? And so this uk transpose Luk is going to be this, which we have already seen. Now, by applying the property that this u is also an eigenvector, you will see that Luk is going to be lambda times uk. And so that lambda can be brought out, lambda k times uk transpose uk. If you also normalize all these vectors so that they are of unit length, then this uk transpose Luk can be shown also to be equivalent to lambda k. Okay. So we have these two equations here. This uk transpose Luk, one we have already shown that this is equivalent to this objective function that we want to minimize. And we have also shown that by the fact that this uk is a unit length eigenvector with a corresponding lambda eigenvalue lambda k, we have shown that uk transpose Luk is lambda k. Okay. Now, if we want to minimize our objective function to reveal the clustering structure in the data, then we better choose a u for which the lambda is also small, right? And that is the reason that spectral clustering chooses the smallest a plus 1 eigenvalues and the corresponding eigenvectors of the Laplacian L. Okay. So if lambda k is small, then the score assigned by uk is such that you will have a nice clustering structure. Large values of aij will lead to small difference between uik and ukj. Okay, ujk and uik. So then that will reveal these two clusters nicely. Okay, so that is the idea. And then that is the idea behind this algorithm. There are some properties that I'm going to skip, but all of this can be formalized There is this nice tutorial by Luxembourg on spectral clustering available here at this link. And it was also published in Statistics and Computing. But the algorithm taken from that work is basically this unnormalized spectral clustering, which is shown here. You construct the similarity matrix like this A matrix here that we have shown before. You compute the Laplacian and then do this dimensionality reduction, so that if you want K clusters, you look at the top K, the smallest K eigenvalues and their corresponding eigenvectors. You stack them together into this matrix U, right? So you have this, let me see. This is this matrix A, also called W in this pseudocode. The similarity matrix, that is an n cross n, where n is the number of cells, right? And then here you have become n cross 3. This is the U matrix. You're looking at these smallest 3. Here we want 3 clusters. So you're looking at the smallest 3 eigenvalues and the corresponding eigenvectors, and then you put these eigenvectors as 3 columns of this matrix to get U. Okay, so that is what is shown here. So you have input as a similarity matrix, which is n cross n, right? This similarity graph, and W is the adjacency matrix, weighted adjacency matrix of this. You compute L equals D minus A, right? You compute the first K eigenvectors U1 to Uk of L if you want K clusters. Okay, so this is U is going to be n cross K. Okay, in this case, n cross 3. By the first K eigenvectors, you mean the K eigenvectors with the smallest K eigenvalues. And we have just seen why it is desirable, because it will reveal the clustering structure in the data. Large Aij will give you very small values of Ui minus Uj. Then, then you apply some other clustering algorithm, because now it has been converted from this more complicated data set into this simple looking reduced dimensionality data set, where any clustering algorithm, reasonable algorithm, will work. And then you can use something like k-means or Leuven or any clustering algorithm of your choice, a simple one. Then you apply on this reduced dimensionality data, and it will reveal these 3 clusters. Okay, if you apply k-means on the original data, you would not have found it out. But on this reduced dimensionality data, k-means would be able to find these 3 clusters. Okay, so that is what is shown here. And then you cluster the points, these yi and r to the k, with the k-means algorithm. So each of the points is now the rows of this matrix n cross 3. You now cluster it, you will get your solution. Okay, so that is the spectral clustering, which is this first step, which is dimensionality reduction using this eigenvectors or eigenmap of your Laplacian. That's why it's called Laplacian eigenmap is the first step. And then a second step, which is a simpler algorithm, k-means or Leuven or some other algorithm, to do the clustering in this reduced dimensionality space. Okay, so that brings us to kind of the main technical part having covered. So what we have seen so far is we have looked at what all the different brain cell types are, and then we wanted to see whether we can find them or find cell subtypes in a more data-driven fashion. And we have argued that looking at the molecular content of the cells will help. And then such data sets are huge and noisy, and after some pre-processing, you have a nice cell cross-gene matrix. You can apply spectral clustering, which involves a dimensionality reduction step to further cluster the data. And then we can apply it recursively to get cell types first. And then within each cell type, we can again apply it to get cell subtypes and so on. Okay. An application of this approach, not the exact same methods, but something very similar, has been done on a particular study on a single cell analysis of Alzheimer's disease. And that's what we will look at next. Okay. Okay. Without further ado, let's just proceed forward. So this is a particular study published in 2019 by Mathis et al. From Manolis Kellis and collaborators and co-workers group. So this is looking at single cell analysis of data collected from 48 individuals. Half of them are they are from the post-mortem brain samples of half of them who died of non-demented cases, non-demented reasons. So these are control individuals, and another 24 who had Alzheimer's disease with different levels of severity. And from them, single nucleus transcriptomes, meaning the gene content of these different cells of 80,000 cells from across these patients, were collected from the particular Brodmann area called 10, which is this frontal pole area of the prefrontal cortex. Okay, the prefrontal cortex is one of the regions that is affected in AD. It is a seat of cognitive function, so that's affected in AD here. Here is the study design. As I said, there is no pathology, 24 postmortem brain samples, and another Alzheimer's disease pathology, a very common form of dementia, very debilitating disorder, and progressively it gets worse that you even forget some basic functions that you used to do as an adult. Okay, so there are the female male is also balanced here. And then you look at this region of the brain, this prefrontal cortex region, you isolate these different cells from these regions from these different patients, and you do this droplet based single nucleus RNA-seq, or the single nucleus transcriptome, right? And this is all the patient cells all pulled together and viewed in this reduced dimensionality space. I mean, they're not using spectral clustering to show these two dimensions. They're using a different method, but the spirit is the same. You're reducing the dimensionality. Each cell will have like 20,000 values, but then you cannot see it. So you just reduce the dimensionality to 2 so that you can plot all these cells in a two-dimensional plane that you're seeing in the screen here. And what is this bar like bar refer to? I mean, these different cells are colored over here. So these are excitatory neurons, about 50% of them. The next largest seems to be oligodendrocytes, right? 26% of them. Then the precursor cells or progenitors of oligodendrocytes, the ones that become oligodendrocytes, these are the cells which form the sheath around the neurons, right? The axons. These are these 4%. Then there are inhibitory neurons, which are like about 13%. And then you have astrocytes, which are the star-shaped cells, about 5%, microglia, 3%, and so on, right? This is what came up in their in their unbiased data-driven scan, right? And there is, of course, bias in the sense that certain cells may be easier to isolate their nuclei than other cells, and so this distribution is affected by certain experimental bias. But when I mean unbiased, I mean the whatever the data tells you, right? And there are these endothelial cells and other types of cells, the blood vessel cells and other types of cells as well. Okay. And all of this coloring has been done by using this clustering approach, using a very similar approach to what we said, not the exact same approach, but a very similar approach. They construct this kind of a similarity matrix shown here, where you have a cells cross cell, N cross N matrix, right? And then you apply this K nearest neighbors to construct a sparse similarity graph. So here is the full similarity graph shown here, but then you remove certain edges which are very sparse by using this K nearest neighbor approach. And before that, you also do certain other dimensionality reduction techniques, right? So, but not looking at the details, the overall rough framework of things is the same. So you just look at the similarity matrix. You use a K nearest neighbor graph to figure out a similarity graph among all these cells. And then you reduce it down to fewer dimensions using a spectral decomposition. In that particular case, they were looking at 15 dimensions. In previously, we used K equals 3 here, right? This is the example that, by the way, these figures and the intuition behind what does this, why do we choose the A+1 smallest eigenvalues? All of these are from slides borrowed from a colleague, Harish, in my department, from his machine learning course. Okay, and basically, so whatever we argued here, we discussed here, right? Similar steps are used, not the exact same steps. The details change. So that you first start with the similarity matrix, you sparsify it using a K nearest neighbor approach, then you combine, then you apply a spectral decomposition of that, then you apply a Lewin method instead of K-means to do the final clustering. And that gives you this pre-clustering, which is the major cell types. Now we can apply this another time, the exact same procedure to each of these cell types to get further subclusters or cell subtypes. Okay, this is what I mean by you not only find cell types, but you also find cell subtypes. Okay. Okay, so what are the findings that come out of these single cell analysis of these Alzheimer's individuals versus control individuals, right? One is that you get a frequency of these different cell types as well as cell subtypes. The cell subtypes are shown here. You can see that the subjects are over here. So this is a subjects cross cell subtypes matrix. So it will tell you in this particular subject, if you look at one column, it will be one subject. Okay. And then that you can see that EX means excitatory neurons, IN means inhibitory neurons, OLI means oligodendrocytes, OPC means oligodendrocyte precursors, and so on, right? So you have these subtypes, about 11 or so, of excitatory neurons, and you have some about, again, in the tens of inhibitory neurons, and you have some subtypes of microglia, 3 or 4 subtypes of microglia, and so on. So you can get the frequency or the senses of these different cell subtypes in the different subjects. Then you can compare between Alzheimer's individuals and control individuals, which cell subtype frequency changes, right? So that is the kind of analysis you could do. You can either do it at the cell level. You can look at, okay, there is this astrocytes is 5% overall in all these individuals, but between Alzheimer's individuals and control individuals, is there a significant change in the frequency of astrocytes and the composition of all of the percentage of how many, which fraction of these cells are astrocytes? Or you can also look at what are the subtypes of astrocytes. Is there any subtypes changing? And indeed, they found many different subtypes and cell types whose frequency changed between Alzheimer's individuals and neurotypical, I mean, controls, right? Control individuals. So that gives you a very finer look at which cells and cell subtypes are changing between these individuals. And that is not the only thing, right? So you can also look at the genes within these cell types which are changing, not just the frequency of cells or cell subtypes which are changing between the disease versus controls, but you can also look at what genes are changing, okay? So here is some example genes, okay? So you have this brain, and then you are showing this microglia, this oligodendrocytes and their precursors, astrocytes, and so on, excitatory and inhibitory neurons. And you can see here that GFAP was something that we already saw before. We looked at the protein view of GFAP, and it was a marker protein for astrocytes, ASTs, right? And this also comes up in this data. So there are all these genes which are more or less enriched or exclusively expressed in astrocytes compared to these other cells. Some of them are known from literature like GFAP, others are found from this data, and all of them may not be fully exclusively, but at least mostly they are present in astrocytes, but not in the other cells. And so these are all marker genes for these different cell types. And we can now inspect if the other genes within these cell types, right, are changing between the disease or not, right? And so once you have an understanding of these marker genes, which are now verified in this data-driven fashion of these different cell types, you can also look at which genes are dysregulated in Alzheimer's disease. What do I mean by dysregulation? So these genes are changing in their average expression, right? So let's look at this particular gene. So blue color means it is increasing in Alzheimer's compared to red, which is decreasing in Alzheimer's, right? So you can see here that this gene called SPP1. So let me give you the legend for this. The rows are all the genes, okay? And this is your snRNA-seq, and this is your bulk RNA-seq. So this is showing the value of the single nucleus, single cell investigation. The bulk is you just take out the tissue and then you just grind it up, and then you look at the average expression. And then if you look at which genes are changing, SPP1 is one particular gene that is changing. It is changing in the average expression level between disease individuals versus control individuals. Okay, so then this will point out to which gene should be looked at for involvement in the disease, for involvement as for use as a biomarker for a disease, or as therapeutic targets for the disease. Okay, so all of these genes are valuable findings in that sense. And then there are some other genes which are changing in the opposite direction, like ICA1 in the bulk sample. Okay, so some of these are changing and are detectable in the bulk sample, some of these genes, but others, such as the ones shown here in microglia, right? This RORA, SIL3, and then RPS19, C1QC, all of them are changing in microglia alone, because the single cell analysis allows you to separate out microglia and then look at microglia of Alzheimer's patients versus controls and look at which genes are changing. But that is not visible at the bulk level, right? At the bulk level, only some genes are visible, and they are mostly those that correspond to the predominant cell types. So here, these are corresponding to oligodendrocytes, and these are corresponding to excitatory neurons. And I just mentioned you that excitatory neurons and oligodendrocytes are the two largest set of cells. So the bulk data is dominated by these most abundant cell types. And so whatever change is happening in the disease in these smaller things like microglia are lost in the bulk data. But then the single cell data reveals those things, such as this stretch of these final set of genes that is not present in bulk, but then you can see it in the microglia right? So what it shows is that it shows the genes within each of the cell type that are changing in AD, and again points to these new genes that would have been missed by standard bulk studies. Okay. So that brings us to hopefully the value of a single nuclear or a single cell analysis that convinces you. So what we have seen so far is that in this lecture is that the modern neuroanatomy has harnessed recent developments in single cell technologies to really revolutionize how you map cell types and cell subtypes in the brain. And that can be applied to both healthy postmortem brain samples as well as deceased brain samples. And the that is what the experimental or technological advance that has been harnessed by recent neuroanatomical research, right? And ML methods have played a crucial role there also, because these are large data sets with lots of noise in them, and so several statistical analyses are done to pre-process the data, to remove the noise, get it into a form like this genes cross cells matrix form. And then the analysis doesn't stop there. Then we need other unsupervised ML methods such as dimensionality reduction and clustering to actually identify these cell types and cell subtypes. I hope I also conveyed that in this lecture. And as a illustration of how this combined experimental computational pipeline is used, we have shown a particular study which has analyzed Alzheimer's disease, and it has revealed not only the frequencies of these cell types and cell subtypes, but also what is happening within these cell types, what genes are changing within them in disease versus controls, which is not visible in a bulk average measurement. Okay. And so looking ahead, I would just like to leave you with this quote by basically a poetry, a haiku by a poet named Arisha. There are so much research that has happened, and some of which I have like just briefly highlighted here, so many more, but then so many more yet undiscovered, right? And the brain is still the most mysterious organ, most complicated, intricate organ, and we do not know all its parts and all its connections yet. And we are performing some single cell or single nuclear RNA-seq experiments on the brains of different species, not just humans. We are planning to do it. So we have a center and funding to do it, and we are planning to do it to study brain evolution. And we hope data will come out of that. And so this link will give you what our current plans are and how we are proceeding with that. And more importantly, looking ahead, what you can do, right? So this is at the whole course level, not just for this particular lecture. So this course and this lecture in particular interleave several threads of neuro and ML tracks. So you should really consider taking the time to reflect on them, see what unique contribution, any form of contribution that you can bring to this very interesting and exciting interface of neuroscience and machine learning. Some of the examples that I showed before are actually written out in these popular press coverage. Like I said, can we understand, can we design better artificial neural network architectures by not just looking at the neuron as a basic unit of processing, but also other cell types that we saw in this lecture, right? That is one particular thing. Can we look at all the brain cell types, and can it inspire new ANN architectures, new artificial neural network architectures? And the other thing is, can you design better machine learning methods to understand novel cell types that have been missed in current approaches, and more efficient methods that can analyze large volumes of data that are being generated now and will be generated in consortiums to come? So these are just some of the few questions. And there is, of course, the question about can you better design power efficient machine learning networks? I mean, brain is, despite all the similarities that we try to show, analogies that we try to show between the neuroscience aspect and the machine learning aspect of the course, they are still very different things. So machine intelligence and human intelligence in how it is achieved. And one biggest difference is in the power consumption, right? Brain is if you will, coarsely a 20-watt device, right? But then it is dwarfed in comparison to how much the data center is required to train a transformer model, let's say, a latest deep learning model. There is this article which says that it can take as much as the carbon footprint and the power hour energy consumption as 5 cars in their whole lifetimes, right? So designing power efficient machine learning methods, machine learning architectures. Those are also really new areas, and I hope you can utilize what you have learned in this course to go understand the context from these popular press coverage, but go beyond it, use the material learned in this course to really understand the primary articles behind these discussions and bring your own contributions to this field. Okay, with that, I will thank you. It has been longer than what I expected, I believe, but hopefully valuable. And thank you for your attention and interest and your time. Thank you.